Next speaker will be Emma Bunting from the Huntington Disease Center, Department of Neurodegenerative Diseases at the Institute of Neurology, UCL London, in the lab from Sarah Tabrizi, and she will give a talk on MSH3-Targeting antisense oligonucleotides halt CAG repeat expansions in HD IPS-derived neurons.
So thank you for the introduction, and thank you for inviting me to speak. So my PhD project has largely focused on the use of an MSH3 antisense oligonucleotide from Ionis Pharmaceuticals, specifically looking at its effect on somatic expansion in Huntington's disease medium spiny neurons. So MSH3 has been identified as a genetic modifier by multiple genome-wide association studies. But what's really exciting about MSH3 is that unlike many of these other DNA repair genes that have been implicated, it's known to tolerate a high level of loss of function variation, meaning there are a lot of people who are walking around with loss of function variants who are completely fine. And that goes hand in hand with mouse data, whereby MSH3 deficient mice have a normal lifespan and are not typically tumor prone. So targeting MSH3 appears to be safe. Additionally, MSH3 has been shown to influence a wide range of repeat expansion diseases, meaning that therapies aimed at lowering MSH3 may have benefit not just in Huntington's disease, but in other repeat expansion disorders. So MSH3 influences somatic expansion in multiple repeat diseases, which is how it's thought to exert its modifier effects. And it does this by recognizing these loop outs that are formed by the repeat tract, recruiting endonucleases, which then cleave the DNA, and it's a downstream repair that then leads to somatic expansion. So if we can lower the levels of MSH3 expression, then we may be able to slow the rate of somatic expansion and prevent that CAG repeat from reaching a length at which it's toxic to the cells. So the aim of this study was to determine the level of MSH3 knockdown that's required to slow or stop somatic expansion in a 125 CAG Huntington's disease iPSC-derived Lyme. So we make MSNs from these iPSCs over the Arbor et al. Protocol over 36 days, and we then treat with the ASO when they reach maturity at doses aiming for 25%, 50%, and maximal non-toxic knockdown, and then we measure repeat expansion over time. So this is just to show that with increasing MSH3 ASO, we get dose-dependent knockdown, and we used this dose response to impute the doses that were required to achieve 25% and 50% knockdown. So as you can see, with dose-dependent MSH3 knockdown, you get a dose-dependent lowering of somatic expansion. And what was really exciting was that we found that with maximal MSH3 lowering, which was about 80% lowering, we actually got this trend towards repeat contractions, although that difference was not significant. So a key question here is, are these contractions real or not? So it could be that it's a selective viability effect, whereby alleles with a shorter repeat length actually survive preferentially. So we tested that using Alamar Blue assays, and you can see that at 9 weeks and at 15 weeks, there is no adverse effect on neuronal viability with ASO treatment. So small contractions are also difficult to distinguish from PCR slippage, so we're also pursuing single molecule CAG sizing techniques in order to further demonstrate that these contractions are real. So we then repeated these experiments in FAM1 knockout MSNs, and the reason for doing that was actually threefold. So firstly, FAM1 knockout cells expand a lot faster than FAM1 wild-type cells, so that gives us a system where we can better detect differences between treatment conditions. FAM1 is also the most significant modifier of onset, so if MSH3 lowering works in FAM1 knockout cells, then it should be effective even in individuals with the highest level of polygenic risk. And thirdly, FAM1 is known to protect against somatic expansion. So there's a key question there, might it also be involved in driving contractions? So what we found was that with maximal MSH3 lowering, the trend toward contractions with MSH3 ASO treatment is actually lost. But importantly, with maximal MSH3 lowering, it still reduced somatic expansion significantly by threefold, even in this highly unstable system. We also looked at the effect on these large, rare repeat expansion events, which are undetectable by fragment analysis. And we found that in our FAM1 knockout system that we actually had spontaneous large expansions occurring all the way up to 403 CAGs, as well as all the way down to 86 CAGs. And we found that with the MSH3 ASO treatment, that actually suppressed the frequency of these large, rare expansions occurring, although these differences were not significant. So in summary, the MSH3 ASO successfully lowers MSH3 in Huntington's disease neurons. MSH3 lowering has a dose-dependent effect on the rate of repeat expansion. Maximal lowering may cause repeat contractions, but we're working to validate that. The MSH3 ASO is still effective at significantly reducing somatic expansion in FAM1 knockout MSNs, and the ASO may suppress the frequency of large, rare expansion events in these MSNs. So finally, I'd just like to acknowledge all of the people that have contributed to this work, including in the Tabrizi Lab team, the Bunting group, people at IONIS and CHDI, as well as all the funding bodies and the patients who have made this work possible. Thank you.
Thank you very much, Emma. A question from the audience?
May I? Sorry? Okay.
Oh, yes, yes, go ahead, and then we can
Terrific. Emma, thank you for that. Paul D'Souza, down here.
Hi.
Perhaps you mentioned it, and forgive me for missing it. How are you stimulating somatic expansion in your terminally differentiated cells, and thereby evaluating the effect of your ASO?
So these are juvenile Huntington's cells, so they have 125 CAG repeats, so they actually expand already in culture robustly. As they are. They don't need stimulation.
Can you help me understand what actually is taking place? Is it oxygen-free radicals in culture that are then, just basically stimulating spontaneous somatic expansion?
I feel like the exact mechanism is actually not definitive, but it's these loop-out structures that form spontaneously with an expanded repeat that then is recognized by MERS beta. And it's then the downstream mechanism, again, is also debated. Okay. But that's what's driving the erroneous repair, yeah.
Okay, thank you so much.
Darren has a question.
A related question in terms of, have you actually measured cell division rates in the cultures? So they are terminally differentiated and non-dividing?
So our cultures are 90% neuronal, and about 10% of them express Ki67. So there is still some proliferative cells in that culture, but we're actually setting up a collaboration with Professor Steve McCarroll, who's able to do single-cell CAG sizing as well as RNA-seq in the same nuclei, so you can tell exactly what's happening in the proliferative cells versus the neuronal cells, and with ASO treatment, knowing which ones the ASO is actually successfully lowering MSH3 within too.
Great, fantastic,
Yeah.
Thank you.
Thank you very much, Emma. In the interest of time, we have to move on.